Hi there, I'm Dennis Province from the IDEA National Resource for Quantitative Proteomics, and I want to talk to you about protein mass spectrometry today. So what are you going to learn today? I'm going to learn a little bit about the resource and what we do, and some discovery proteomics, the workflow that we call discovery proteomics, and then I'm going to use an example to talk about design, how we collect the data, and how we analyze that data. So the IDEA National Resource for Quantitative Proteomics mission is to provide state-of-the-art proteomic services to nationwide user base. And we do that by having instrumentation, sample prep services, and bioinformatics. And we also provide educational opportunities like workshops, symposiums, and also outreach and training like voucher programs and internships. The grant is written by Alan Tackett, and our funding goes through 2030. This is a map that shows where we get projects from all over the country, and the bulk of those projects coming regionally since we started there in Little Rock, Arkansas at this core almost 10 years ago, but we've been a fully nationwide resource for 6 years now. Here's a group of people that I work with: bioinformatics, sample prep, instrument specialists, administrators. A lot of people make up our team, and it's all necessary for us to do the job that we do. We're located in the University of Arkansas for Medical Sciences campus in the Cancer Institute on the 9th floor, and that's in Little Rock, Arkansas. And we do discovery proteomics, which essentially means we try to identify and quantify all proteins in a sample. So you've learned a little bit about us. Let's talk about discovery proteomics. Proteomics is the study of proteins, but you didn't see that coming. If that's true, then genomics would be the study of genes, and metabolomics would be the study of, that's right, metabolites. Discovery, if you put discovery in front of that, that means we don't know exactly what we're looking for. We've designed some sort of experiment to cause a change in the proteome, but we're not exactly sure what's going to happen as a result of that. We're not looking at just one protein or one set of proteins. We want to see how this change affects the proteome globally. So this is a picture here that shows the gel bands of a helolysate. And just to kind of remind you, let me get my pointer going here, that if you run an SDS page on a protein lysate, and as the proteins migrate down this gel, and then you stain them with Kamasi blue, you'll be able to see these different bands here, which represent different proteins at different amounts as we come across here. So all of these proteins together could be the proteome. And you might be interested in doing proteomics on a single band, which is actually probably hundreds of proteins all included in that one band. But if you've done gel work before, you've done a Western blot, you've done proteomics. What I'm going to talk about is protein mass spectrometry, where we use a mass spectrometer to do our proteomics. So what is the proteome? Well, the entire set of proteins expressed by a genome, a cell, tissue, or an organism at a certain point in time under certain defined conditions. Once we stop that reaction, we form a lysate. All of those proteins then measured at one time is what we call the proteome. And that can be in cells, that can be in tissues. And there are over 80,000 different proteins and hundreds of thousands of variants and modifications. So your basic biology might have told you that, simplified it and said that we have information encoded in our genes that gets transcribed into RNA, which gets translated into proteins, and you may have thought that one gene will equal one protein. That's not true. There's only about 20,000 genes, but there's about 80,000 different protein variants of that because of some different modifications that can happen Well, what is a protein? To go down a little bit further, the chain of amino acids connected together like beads on a string, where each one of these beads is a different molecule, and the side chain of that molecule represents what amino acid it is, whether it's alanine or isoleucine, etc. And so the primary sequence of amino acids is the beads or the amino acids connected together through this amide bond, but then, as you know, as you go on, that the type of side groups there will cause it to fold up in different structures here, and the structure is actually what causes the function. Well, if we were to look down even further about the chemical structure of the protein, and if I'm just looking at a small swath, about 25 amino acids of the amyloid beta protein here, we have an N terminal and a C terminal, and then you've got that amide backbone coming across here, and you can see these different side groups coming off. You can represent that chemically, showing all the bonds here, like you do in this picture, or you could also use a single digit code or a 3-letter code to represent just the amino acids. And for instance, this guy right here, that's the side chain for tyrosine. And where I have marked these little red lines, that would be where comes a little bit later, but if we were to cleave or cut up this protein into smaller pieces, if we could use an enzyme that specifically cleaved at the C terminal of certain amino acids, then it would cause this large protein to be broken up into smaller peptides. Well, that's what bottom-up proteomics is. Bottom-up proteomics is where we're going to study the proteome. We're going to look at all the proteins in a sample, but first we're going to take those proteins and we're going to digest it into peptides using a proteolytic enzyme. So proteins are really sticky. Think of making scrambled eggs. The proteins are really long and they tend to clump together, and they're very difficult to analyze. It's very difficult to make proteins into a solution. And if you can't make it into a solution, it's hard to dilute, it's hard to transfer. But peptides, on the other hand, that would be having only 7 to say 20, maybe 30 amino acids in length. They're much easier to analyze, especially in a mass spectrometer. So we're going to take a protein, we're going to take a proteolytic enzyme like trypsin that will cleave specifically. So trypsin, you might think of as a giant pair of scissors here, and it's going to take that protein and cut it up into very specific pieces that we call tryptic peptides. And that's what we're going to actually separate by liquid chromatography, and then we're going to analyze in our mass spectrometer. Well, how are we able to separate those peptides? We use this really fancy thing called liquid chromatography. And the goal of liquid chromatography is to separate the peptides into distinct groups. So we want to separate them by their hydrophobicity. More hydrophilic, which are the smaller, higher charged peptides, will elute early. And the hydrophobic ones are larger, they have less charge, and they'll elute late. And the goal of chromatography is to crowd those peptides together into concentrated groups that can be analyzed individually. So you might think of a chromatographic system to do this. It takes a bunch of peptides that are all jumbled together, and the process is going to cause those peptides to break out. And of course, my little picture here is covering them up, but it separates them into the green and the purple and the yellow and the red peptides that are represented here, so that they're all concentrated together. Well, how do we detect and identify those peptides? Well, first of all, peptides are positively charged. They usually have a +2 or a +3 or +4 charge. Peptides have a specific mass, and anything that has this charge and a mass can be measured using a mass spectrometer, because mass spectrometers measure mass to charge, M/Z. If we measure if we run a scan, we'd call that the MS1 signal. And then if we can take one of those signals, we can fragment one peptide, one particular type of peptide, into smaller fragments. We call that the MS2 signal. So those are the two basic pieces of information that we're looking for. So for instance, that green peptide that we just separated by chromatography, we're going to hit it with some energy. It's called collision-induced dissociation. And it's going to cause fragments to form of that green peptide. So each one of these little peptides here is a fragment of the larger green peptide. And then from those fragments, we can get signal. So what does the initial data look like? So if we're running a lysate from human cells or some sort of organism like that, we could have 100,000 peptides in a single sample. It's for a 30, 40, 90-minute run, something like that. You're going to have all those peptides coming out almost on top of each other. And it's going to seem really jumbled, like a forest of trees in here. The goal is to have these peptides spread out over this time window by this gradient. We are increasing the organic content, eluting those peptides off of the column, and that's causing the early eluters. This would be the hydrophilic peptides coming out first, and the hydrophobic peptides coming out at the end here. Each one of these signals represents a possible peptide. So if we look at that one peptide here and we'd run a scan, we might see at that one point in time, we might see this happen. We might see a group of signals here. So we have M over Z on the bottom and we have relative abundance here. And so these are the signals that all came out at that one moment in time. And each one of these guys could be a peptide. That could be a peptide. That could be a peptide. We want to look at all of these, but especially we want to look at that big guy up there, right? Why? Well, because it's the most abundant. But most likely the peptide that we're really interested in is this little tiny one here that you can't see because of my camera that's really, really small. But we can also analyze those too. So we have a decision to make as we get all of these signals is how do we isolate and send each one of these signals into the mass spectrometer to be fragmented and analyzed. And so there's a trade-off here where because those peptides are constantly coming out, there's a time element here, and the detector can only process a certain number of things at a certain amount of time. You have to decide how deep you want, how much signal that you get for each one. Well, how do we interpret that MS2 spectra? That was what the MS1 spectra looked like. Well, the collision gas breaks the peptide into fragments, and the break usually occurs upon specific points on the peptide backbone. And so if this picture over here kind of represents a peptide where you have the C terminal over here and you've got the N terminal over here, what we find is that if we hit, if we send gas molecules, this is all done in the vacuum in the mass spectrometer. And so if we introduce a little bit of nitrogen gas that has a little bit of energy, or helium gas that has a certain amount of energy to it, and it bumps into these guys, it will break at very specific points. And that would be along this YB place right here, creating what we call Y ions and B ions. And the loss of mass is equal to the amino acid. So here's what my MS2 signal would look like. We still have M over Z, or mass over charge, on this axis, and we have the relative abundance over here. But now each one of these signals does not represent a peptide. There was a single peptide that was fragmented, and these are the fragment pieces of that peptide. And so how we would figure out what peptide we're dealing with is we would take and subtract the difference between these signals to come up with the mass of very specific amino acids. So just to remind you, these green fragments that we talked about over here are going to be these different signals right here. And so if we subtract these guys out, we can decide whether this mass difference here is because of a tyrosine or a glycine or a serine, etc. Well, what type of tryptic peptides do we make and detect? So if I were to take, remember, trypsin is that proteolytic enzyme that will cleave specifically after a lysine or an arginine. And if I take an example here, would be human insulin-like growth factor. And this is the amino acid sequence, all 605 amino acids here, and I were to subject that to trypsin at the right pH and concentration, that trypsin is going to come along and it's going to cleave after every R and every K, or every lysine and arginine in here, to create these smaller fragments, these smaller peptides of this entire protein. Now, some of you at this point, you're going to say, oh, no, hold on. You said there are like tens of thousands of proteins, and now you're going to add this trypsin and you're going to create hundreds of thousands of peptides. Isn't that making our job that much harder? In a way it is, but I'm going to show you how a neat trick that we can do to come back and put all this together again after we make peptides. So some of these peptides are really small, like for instance, this lysine right after this arginine. That's a single amino acid. We're never going to see that. In fact, we probably won't even see this little guy here that has only 5 amino acids. It only has to, but we're going to form 70 different possible peptides here if we don't have any miscleavages just from adding trypsin to this. Now, not all those peptides are going to be ionized. Not all of them are going to be detected. And so there's going to be gaps in the sequence. But the good news is it only takes 2 unique peptides to have a score above the predefined scoring mechanism to be able to declare that we found this protein here. Okay, so we only need 2 peptides, 2 unique peptides that match to this guy. But like I said, unfortunately, a little tiny peptide like this is going to be too small, and probably there's going to be like this guy here, this blue guy here, that one's really big. That's probably 58 amino acids in length. That's probably too long for us to analyze. It will never come off the column. So what does the actual coverage look like for this protein? So after doing a real experiment with samples that have this protein in it, we found this coverage. This is what we call a coverage map, where what's outlined in gold here are the peptides that were actually found from this human insulin-like growth factor. And if we look at just specifically the MS2 signal for this peptide, we get something that looks like this. You'll see over here these matching Y ions and B ions that match to this table. So this table kind of shows you all the theoretical masses that would be calculated by taking a peptide of this makeup and breaking it apart into pieces, into fragments. And then we can, from algorithms, we know the relative heights or intensities of these different fragments in a piece like this. And you can see up here at the top how different fragment pieces that we have, by subtracting that, will give us different pieces of information, whether we know this is a threonine and a glycine, whether those are prolines there, etc., from either the Y ions or the B ions. And so this is like a fingerprint. Each one of these peptides is going to have a MS2 signal, which is like a fingerprint that matches back to that specific peptide. Well, how do we know what proteins that we're looking for? Because of the Human Genome Project that was finished a number of years ago, we have libraries now for all the different organisms. So you can look up on a website called Uniprot, and you can look up in the proteomes and you can type in Homo sapiens, and it will pull up and it will give you the codes for humans. These would be a list of proteins that are found generically in every human cell. And there are about 20,000 of them if you just take the most predominant protein sequence per gene. And if you count all of the variants and then you'll get up to above 80,000 proteins total. So this is one of what the entry looks like. So the library is actually a bunch of these entries that has some information that tells you basically a specific protein number, and then a little description of what that protein is, and then the primary sequence of that protein. And so the computer's job is to identify from the MS2 signal what the sequence is of the peptide, then take that sequence and go to our library and see which one it matches with. And when it's almost like if I took a book like War and Peace, that's a really large novel by Tolstoy, and I were to cut out 8 or 10 words out of a sentence there, and I were to hand that to you, and you had to go through all 1,000 pages of the book and look to find what chapter and what paragraph those words combinations came from. But eventually you'd find it, and that's exactly what we do in a search, in a database search in proteomics. So what have we learned so far? We learned that genomics has provided us libraries of proteins. Those proteins can be cut up into peptides, and the peptides and their fragments can be identified by mass spectrometry. So if I were to put that in pictures, we take the biological information, which are the proteins. We're going to add trypsin to make peptides. We're going to use liquid chromatography and mass spectrometry to get two basic pieces of information, the MS1 signal and the MS2 signal. And then from that, we're going to do a database search where we're going to find the identity of the peptides and then the proteins. And each signal up here in the MS1 represents a precursor or the peptide. And each signal down here represents a fragment of a particular peptide. And so this is what we're doing is somebody's coming to us with biological information. They're coming to us with like tissue samples or cell pellets or a serum or some sort of clinical fluid from the body like blood. And we are taking that information that's in there. We are extracting the proteins. We're undergoing this trypsin digestion to get peptides. We are running those peptides on our instrument, and we are getting digital information in the form of a giant spreadsheet. So what have you learned so far? We learned a little bit about discovery proteomics just now, a crash course, and you learned a little bit about the national resource, and now we're going to talk about an experiment. So how can we use this workflow in an experiment? Well, we need an experiment. We need to have a hypothesis. So what about how does a diet high in fish oil change the heart proteome of a mouse model? Or another way to ask that question would be, what are the heart proteome differences in a mouse model fed with a control diet versus a diet high in omega-3 fatty acids, which is basically fish oil? So we've identified an organism. We're going to use a mouse. It says organ is liver, but it's actually heart. I don't know who makes up these slides. It's hard to find good help, isn't it? There's going to be two groups. There's going to be a control group and there's going to be a fish oil group. And then we're going to identify the number of samples per group as 15. And just in case we're, how are we going to do this? We're actually going to go through what's called differential analysis. We're not really concerned with the actual absolute quantity of anything. What we want to know is how does a particular protein change expression? How does it change as we go from the control group to the fish oil group? Do the proteins go up or do they go down relative to the control? So we're going to do what's called differential analysis, where we quantitate the differences between the proteins in these two groups. So what are the sources of variability? This is important whenever you're doing an experimental design. What we hope is that the variability, that the change that we are actually trying to measure is the biological variability of the food source, that everything else is held the same. But that's a challenge because we're going to have different animals. They're going to be in different cages. They're going to have to do some surgeries to get the heart tissue. We're going to have to take the tissue out and we're going to have to perfuse it, which means basically wash it with PBS to get as much blood out of it as possible. We're going to have maybe some of those animals are going to be male and some are going to be female. That could cause a difference. Some of those animals might be older than others. We would want to design the experiment to control as many of these things as possible so that they do not add additional variability into our experiment. Also, our sample preparation, when we do our trypsin digest, we want to be able to do all of the sample prep at the same time on the same day, the same lot of trypsin. Data collection, does the instrument running correctly? It's going to take, for this experiment, it's going to take over a day to collect all this data. We hope that the instrument will be in the same condition at the beginning of the experiment as it is at the end, and we want to be able to control for that. Also, there was a picture down in the corner of what an omega-3 fatty acid looks like. And that is really the experiment that we're doing is how that particular fatty acid changes our heart proteome. Well, what's the summary of the experiment? So we're going to have two groups, control group and a fish oil group, fed different mouse populations, same sex, same age, same environment as much as we can. We're going to have we're going to have some surgeries on the heart control heart and the fish oil heart. We're going to try to do that on the same day in the same environment, maybe even by the same sets of hands if we can. And that'll be tough to do that many surgeries. We're going to get some control tissue, and then we're going to have to make lysates. We're going to have to break apart that tissue, break apart those cells. Then we're going to have to do a protein quant and a trypsin digest. We're going to have some control samples and some fish oil samples. And then we're going to have to, after a tryptic digest, we're going to run these on the LC system. We're going to want to intermingle these. We're not going to want to run all the controls first and all the fish oil second. We're going to want to randomize how we run these. So that's not a batching. We're going to get all this information, the MS1 and MS2 information for each one of those runs. Then we're going to do a database search. That's where we go through and we figure out what peptides we found and what peptides we have in our library of our mouse library, and we'll come up with what proteins and their abundance that we have here. Then we're going to get these a big spreadsheet that's going to have we call the protein results file, and then from that we're going to do our QC and our differential analysis. So how does the database search work? So we're going to take the chromatogram runs from each sample. We're also going to take that proteome that we talked about. And before I talked about the human proteome, but we're not going to use that one. We're going to use the mus musculus because we're doing heart samples from mice. And so we're going to find the library that's specific for mice, has and we're going to download that, and then we're going to use some searching software. And the software we did for this experiment was called Spectronaut, which is a commercially available software that will go through and match these things up and give you an output. Well, what does the data output look like? Well, I've cleaned it up just a little bit just for this presentation, but essentially you have different parts. You've got what we call the annotation data. That's going to be the protein accession numbers. You've got gene IDs, and you've got the protein descriptions over here. You've also got maybe some information that you want, like for instance, it might be helpful for you to know how many peptides were detected per protein. That can give you some idea of the strength of that identification. And then you're going to have some abundance information for your control, and you're going to have some abundance information for your fish oil group. And I've only shown you 3 of each, but remember, we've got, and I'm only showing you for the first couple of proteins here, but we have almost 4,000 protein groups that we found in this experiment. And remember, we have 30 total samples over on the other side. So it's a great big spreadsheet. So before we do our analysis, what other files do we need? Well, we need a metadata file, and this is where we link the real sample names to the group names. Like for instance, when we ran it, we actually used the last name of the investigator and a date, and we have some other information here that you'll find is actually specific to the animal. And so there's a lot of this file name here may not mean anything to you, but what's important is that this file name is in the control diet group. And this is the sample name that can go back to the person that actually did the experiment, because the person that did the experiment with the mice and the dissection and the surgeries and all that is a different person in a different place that runs the experiment on the mass spectrometer. And so you got to make sure you have all of this data linked. The other thing we need is a contrast file. We need to decide what are we contrasting, and this one's easy. We only have 2 groups. We're going to do the fish oil diet versus the control diet, which means we're going to subtract the control diet from the fish oil diet. It's important which way we put these, kind of like subtraction. You have a you always want to make sure you subtract in the correct way. We're going to use a package, an R package called ProteoDA to run what's called Lima, and it's basically like a least squared analysis, kind of like a t-test for thousands of things all at the same time. And I'm just going to run through this, but this package was developed by some people that worked in our lab a few years ago. It was spearheaded by a bioinformatician named Stephanie Byrum. And what this package does is it separates the annotation from the intensities, and it uses the metadata file to assign the groups, filters out samples with missing values. We want to make sure that we have that if we have missing values, if we that at least 2/3 of the sample in one group has to contain the protein for us to count it. We want to write a normalization report. We want to evaluate that. Then we want to write a QC report. And then we want to add a model design, and we want to write those lemma tables that does the actual differential analysis. And then we're also going to do a volcano plot and an MD plot that'll give us something visual to look at. Well, what do those QC plots look like? So one of the plots we have is called a violin plot. And so this shows for the samples that we have here, the blue are the control diets and the red are the fish oil diets. These have, it's like looking down on the data from the top down. So it's like you would expect your data to be Gaussian in nature. It's going to have, most of the data is going to be centered in the middle, and you're going to have a little bit of data at the low end and a little bit of data at the high end. And so this black line represents the mean here, and you can see that it's pretty good for tissue sample that we've normalized pretty well on the front end. We did a protein quant on the front end to say we want to start with about the same amount of protein for each sample, and I think we did a pretty good job. So we hope that this violin plot, everything looks the same. Is the load the same? That's what we're looking for there. For the PCA plot, which stands for Principal Component Analysis, this is essentially taking all of your data and it's saying, okay, let's look at the top 500 most variable proteins and see how these samples shake out. And what you notice is that the red is the fish oil diet, and the fish oil diet samples tend to migrate over on this side, and the control diet samples tend to migrate over here. That's fantastic because the biggest change that we want is due to our hypothesis. And our hypothesis is there's going to be a difference in the heart proteome between the control diet and the fish oil diet, and that's what we see. The next biggest change is going to be in this vertical direction here, between the samples that are up here and the samples that are down here. Well, what does normalization do? Normalization removes some of the technical variability, hopefully, without removing the biological variability. So if, for instance, if there was a huge discrepancy in the types of animals that we used, or their age, or there was a maybe some were male and some were female, or something like that, the normalization, that's going to cause that's actually, some of that's going to be batching effect. But if there's some technical variability, maybe due to the surgeries or something like that, that's causing in-group spread, normalization will kind of tamp that down a little bit. And so this is what this graph shows you, the log fold change of the comparisons here for each dot represents a protein. And this is the intensity down here. And so you have the most intense proteins, abundant proteins on this end, and the least ones here. And if you draw a best fit line, you see that best fit line is not quite through zero. And normalization is going to try to adjust this line by squeezing together the data just a little bit to try to make this best fit line more parallel. And so, for instance, we chose a normalization process called VSN, which normalizes the variability. But you can see that these look very, very similar because the normalization, there wasn't much to be normalized here. Well, what's the normal way to evaluate omics data? So we said this is proteomics data versus genomics data or whatever. So how do you do an omics? Well, first of all, we do this log 2 fold change. Has to be greater than 1. So we're looking for the difference. When we look for the comparison between these groups, we want to see a log fold change of 1. That's just kind of a starting off point. And we want to look at the adjusted p-value, which has something to do with how much the data is spread in group, that value has to be less than 0.05. Well, both of these must be true for a protein to be different in the fish oil group and the control group for a small sample set. But with 15 animal groups, right? So if we just had, so what I'm saying here is if, let's say we did this and we only did a 3x3. We only had 3 control animals and 3 fish oil animals here. we're going to want to be much more stringent and stay to this. But because we had 15 animals per group, I can do some side calculations and find out that I can actually reliably see down to 0.3 fold change instead of a full change of 1. So this is what's called a volcano plot. This is the graphical way to view the data. And this shows you the log fold change on this axis here, both in the positive direction and the negative direction. It shows you the adjusted p-value on this axis over here. And the red and the blue dots represent the proteins that are either upregulated when the fish oil is compared to the control or downregulated. And so the best way to look at this would be to evaluate this by, first of all, seeing which ones are the most significant proteins, right? Because there are almost 4,000 proteins here. That's a lot of data to look at. So what we want to do is we want to look at the data that's the most interesting to us. So we probably want to start with the things that are out there on the edges. We want to start with the significant increases in this direction. That would be where the abundance is greater in the fish oil group than in the control group, or significance increases in this direction, right? And the greater abundance is in the control group there. And so we would want to go through and click on some of these guys and look at those and be able to evaluate, are these particular proteins somehow related to this experiment that I've done? And if so, how? The biology of having fish oil in a diet is that it changes the heart proteome. Well, how does it change the heart proteome? So one of the things I want to look at is that is if you go back to pathway analysis, long chain fatty acids tend to undergo this pathway where they use certain proteins that are needed to be able to take that long chain fatty acid, which is what your omega-3 fatty acid is, and break it down into smaller pieces, and it needs lots of these proteins to do that. So one of the things we can look at is say, hey, are these proteins that are listed in blue here, these are different gene names for proteins, are those guys upregulated when we feed these mice a prolonged diet that includes a lot of fish oil? And these proteins come from the mitochondria. The mitochondria is where, in these different, this little cartoon here just shows some different layers in the mitochondria and where some of those proteins are located that then are used to break down the long chain fatty acids. So all of those proteins that I just showed you there are all listed right here on the positive side of this a volcano plot. And if I look at the log fold change of all of those guys, with the exception of these two, all the rest of them have a log fold change that's above or at 0.3, which we said for a 15 by 15 experiment such as this is a very good indication that all of these are significantly upregulated. So all we show, these are like positive controls to say, hey, we expected when we took this fish oil diet and we fed it to this mouse model, that the heart proteome is going to change by the mitochondria having to assimilate more of those long the fatty acid, long chain fatty acids. And so we would expect those proteins to be upregulated because of the change in diet, and we just showed that. Well, what do these results imply? Well, if you take those proteins and you go start looking for what other pathways they belong to, you start to get what are called mechanistic interpretation. You get strong peroxisomal proliferation and remodeling. That means that the heart tissue in the fish oil diet group is actually changing. You also get an enhanced very long chain fatty acid oxidation, which we showed that all those proteins and those pathways were upregulated. You get coordinated mitochondrial fatty acid oxidation increase, lipid handling proteins increase, and possible mild plasma lipid remodeling signature. So all of these things imply that we are, by just making a single change in the diet of these mice, that we're changing the way the heart is structured. So the conclusion is that the heart metabolism structure is changing because of this fish oil diet. Now, why are we interested in that? Well, because this could be really important for Americans. Heart disease is the #1 killer. Can omega-3 supplements help to prevent heart disease and other cardiovascular events? Just this one study is, you might think, man, that's an awful lot of work to do. Did you really have to, take all those mice and take care of them and go through all of this experiment for this? Wasn't there an easier way? Well, actually, this is probably the cheapest and fastest way to get information to allow us to continue on so that we can do more enhanced studies. In 2020, there were 86 studies that involved over 160,000 people that found increasing EPA and DHA. Those are two different types of omega-3 fatty acids. They slightly reduce the risk of coronary events and coronary death. 2019, they had 13 major supplementation studies, including over 120,000 participants, showed that omega-3 from fish oil lowered the risk of myocardial infarction, that's heart attacks, coronary heart disease, that would be like clogging of the arteries. And then 2019, there was a review of these 3 different trials, concluded that the findings of each study showed that for the general population, consuming 1 gram per day of omega-3 fatty acids provides some benefit to cardiovascular health. And a lot of times the results of these, they tend to downplay it, but there's some significant changes in the heart remodeling that occur because of having an increase of diet of fatty acids. And the reason why fish oil, that's fatty acids from fish oil, and the reason how this all began, in case you were didn't aware of this history, is that there were cardiologists that were studying different populations, and they found that different people groups that are from the far north that have almost a complete diet of fish have little to no heart problems like the rest of us that live off of beef tallow or beef fat or other fatty acids other than from fish. And so that's why a study like this began. So what does all this mean? It means, for one of the conclusions is, I just wanted you to see that chemists and biologists, computer scientists, and statisticians all need to work together on a project like this. It takes someone with vision that wants to work with animals. It takes people that are willing to work with instruments, people in sample prep, people to work with the data on the back end, like computer scientists and statisticians, to make these programs that we can use. And they're all needed, as well as a host of other scientists working together to perform a single experiment to address a simple hypothesis. And at this time, I will take questions. But since this is online, I guess you won't be asking any questions. So I guess I'm done. Thanks for listening. I hope you enjoyed this.